Hi, and welcome to the 52nd Interscience Conference on Antimicrobial Agents in Chemotherapy, otherwise known as ICAAC, an annual infectious diseases meeting of the American Society for Microbiology. I am Jim Sliwa, the Media Relations Manager for the ASM, and I'm here to welcome you to the overview briefing, where we give a brief overview of important topics being presented over the next 3 days. When we reach the Q&A section, those of you who are watching us on the internet can ask questions via Twitter using the hashtag #ICAAC, ICAAC, or can ask questions directly to @ASM Newsroom. We have here 4 members of the program committee for this year's meeting who will be giving us an overview of the meeting. First, we have the program chair, Lindsay Grayson, who is with Austin Health in Melbourne, Australia, who will be moderating. Lindsay, if you would like to go ahead.
Thanks very much, Jim. Well, welcome everyone to the 52nd ICAAC in San Francisco. I'd like to introduce the rest of the panel. So on my immediate left is Craig Rubens, the Vice Chair of the ICAAC Program Committee. Next is Dr. Mike Pucci, also on the Program Committee, and Laurent Kaiser, who's a member on the Program Committee, particularly with an interest in virology. So what we planned to do today was actually kick off with Mike discussing some of the new drugs that are coming online and the new antimicrobial agents, then move to Craig, who's going to discuss pathogenesis, vaccines and pediatrics, Laurent with virology, and then I'll cover the other topics. So Mike, would you like to start?
Good afternoon. As you know, ICAAC has long been a forum for the introduction of new antimicrobial compounds. There has been a shift over the past several years from presentations predominantly by large pharmaceutical companies to smaller companies and academics. And as the large companies have either eliminated or downsized their research efforts. Despite this trend, the number of abstracts submitted this year in the new agents category has actually been slightly higher than last year. In addition, uncertainty in the regulatory environment has dampened enthusiasm for antibacterial drug programs in recent years. However, some positive events have occurred recently that could provide new life to research and development in this area. The FDA has been working to compose and disseminate new guidelines in regard to clinical development of these drugs, while efforts in Europe have dramatically improved the regulatory environment on that continent. This summer, the passage through the U.S. Congress and signing into law of the GAIN Act also adds incentives in the United States in regard to exclusivity, priority review, and fast-track status for antibacterial and antifungal drugs. And this all is encouraging since development issues often impact the research side of the drug discovery process. So this year, we have a number of new promising compounds, albeit at earlier stages of development, and I'd like to highlight a few of these compounds that you will see over the course of the conference. I'll preface this by saying that I have obtained the following information from the poster abstracts and some from corporate statements about their compounds. I'd like to start with 2 compounds that have probably the most presentations here at the meeting. First, Trius Pharmaceuticals has 11 poster presentations describing their new compound class that targets bacterial, JARB, and topoisomerase 4, both topoisomerase enzymes that are involved in DNA replication. These compounds are highly selective for bacterial targets versus other ATP-binding enzymes, including HPS90 and human kinases that you find in mammalian cells. This class is derived from rationally designed molecules that, due to their dual target activity, have extremely low frequencies of resistance thus far and have demonstrated no cross-resistance to other antibacterial agents in use. These topoisomerase inhibitors are effective against all drug-resistant bacteria known, including multi-drug-resistant pathogens such as Enterococcus faecium, Staph aureus, Klebsiella, Acinetobacter, Pseudomonas aeruginosa, and Enterobacter species. In vivo efficacy has been demonstrated in multiple and diverse animal efficacy models, including lung, thigh, kidney, and sepsis, and versus strep pneumonia, E. coli, and Burkholderia pseudomallei, which is a potential bioterrorism pathogen. These compounds have the potential to be the first new broad-spectrum antibiotic class discovered in over 50 years. Rempex Pharmaceuticals is presenting 11 posters describing for the first time the antimicrobial activity, in vivo efficacy, and preclinical safety of Carbovance. That's a new antibiotic combination that is comprised of a carbapenem in combination with a beta-lactamase inhibitor. Beta-lactamase inhibitors are agents that inhibit bacterial enzymes that destroy beta-lactam antibiotics and cause resistance. In particular, Carbovance was designed to overcome gram-negative bacteria that produce beta-lactamase enzymes that have spread rapidly in the US and Europe, including strains of the multi-drug resistant KPC-producing Klebsiella pneumoniae, which has become a very big problem. This combination agent has excellent activity against clinical isolates of multi-drug resistant bacteria in animal models of infection, and excellent pharmacology and safety in IND-enabling preclinical studies. Human studies have been initiated in this program, and the company is currently working with clinical investigators and regulatory authorities on designing a development pathway that can speed the availability of this agent to patients and clinicians. As you probably know, modified derivatives of existing drug classes historically have been important additions to our antimicrobial arsenal, and this year, several such analogs are described. The quinolone class has been successfully used for many years, led by the commercially successful ciprofloxacin and levofloxacin drugs. Daiichi Sankyo has a compound called DS8587 that is a new quinolone analog with broad-spectrum antimicrobial activity, including higher potency against quinolone-resistant organisms. It exhibited potent inhibitory activity against its bacterial targets, possessed greater bactericidal activity than levofloxacin, and was found to have a lower risk to select for resistant variants. This compound is projected for empirical use in both the community and hospital-acquired infections and has begun human clinical trials. Another new quinolone that's being presented is KPI-10 by Calidex Pharmaceuticals, also an inhibitor of bacterial topoisomerases, and has shown broad and balanced activity against a variety of broad panel of gram-positive and gram-negative pathogens, including Staphylococci, Streptococcus pneumoniae, E. coli, Acinetobacter, including strains with the NDM1 enzyme that has made a lot of news in the past year. And this also include many multi-drug resistant and fluoroquinolone resistant strains. Initial data from an ongoing phase 1 clinical study will be presented at the meeting, showing that single ascending doses of oral KPI-10 were generally safe and well tolerated in healthy volunteers with a pharmacokinetic or PK profile that may actually support once daily oral dosing. And the PK profile was found to be similar to that of other fluoroquinolones, and therefore offering extensive distribution into body tissues and fluids and good oral bioavailability. Calidex is developing both intravenous and oral formulations in parallel and plans to advance both formulations through registration studies simultaneously. A more recent class of antibiotics, the oxazolidinones, exemplified by linezolid or Zyvox, has also demonstrated important clinical utility. Efforts have continued to improve the safety profile of this class of compounds. MRX-1 is a new oxazolidinone active against gram-positive pathogens, currently under clinical investigation by ICAAC Pharmaceuticals. It offers greater than a 3-fold reduction in MOA inhibition as compared to linezolid and does not exhibit any apparent toxicity in regard to CYP inhibition, clastogenic, and mutagenic potential assays. And thus far in humans, MRX-1 exhibits high exposure levels similar to those reported for linezolid and is well tolerated and notably attenuated myelosuppression potential as compared to linezolid. And then finally, calling attention to a couple of other types of pathogens, antibiotic-resistant Mycobacterium tuberculosis strains have been increasing over recent years, as we all know, and have made the headlines numerous times this year. A new nitroimidazole, TBA354, is under development by the Global Alliance for TB and Johns Hopkins. This compound is narrow spectrum, highly bactericidal against replicating and non-replicating TB, and maintains excellent activity against clinical isolates, including those resistant to existing TB drugs. And finally, in regard to new antifungal compounds, Astellas Pharmaceuticals describes a novel second-generation echinocandin, ASP9726, with broad-spectrum activity against key pathogens, including Candida species and Aspergillus fumigatus. And these include echinocandin, first-generation echinocandin-resistant strains. The therapeutic efficacy of first-generation echinocandins against invasive pulmonary aspergillosis, or IPA, is currently not optimal. This compound demonstrated enhanced efficacy in multiple animal models against IPA. Because of this impressive activity against aspergillus, compared with first-generation echinocandins, ASP9726 shows promise for improving the therapeutic efficacy of echinocandins in IPA. And I'd like to call attention, there's a session this afternoon at 1:45. It's called the Poster Summary Session for Early New Antimicrobial Agents. It'll be comprised of 9 short talks of new, what we think are promising new agents, including some of the ones I just talked about. That's in room 104 at 1:45. There are also sessions on new treatments for Mycobacterium tuberculosis infections at 4 o'clock today in room 122. There's a Meet the Experts discussion. As I told you, there are new developments in the regulatory environment. So that one is especially concentrating on why there are too few new anti-gram-negative pathogens and how to change the landscape. That's Tuesday morning, 7 a.m., room 122. And finally, on Wednesday, there's a symposium on new approaches to the treatment of fungal infections at 9:15, room 309. And I'll close there.
Thanks very much, Mike. Craig, would you like to?
Yes, thank you. Welcome to the 52nd ICAAC. My job is to highlight some of the things going on in pathogenesis, which is the study of microbes and their human hosts, and the various aspects of what defines pathways to infection or susceptibility. A couple things, I'm going to do this on a day-to-day basis here. Today there's a poster session looking at various factors that lead to infections. One of the most important things are polymorphisms in genes, in human genes, that may make certain populations more susceptible, including if they have underlying diseases like diabetes. A highlight of one of the posters today at 11:30, and this session goes from 11:30 to 1:30 in the poster room, is an abstract entitled Staph aureus nasal carriage might be associated with vitamin D receptor polymorphism in type 2 diabetes. This is a very important study that is now beginning to look at how mutations in the vitamin D gene may actually make those patients with diabetes mellitus more susceptible to persistent carriage of Staph aureus that is a common pathogen in these patients. A second poster presented there today is on the recognition of pattern recognition receptor polymorphisms and the susceptibility to complicated skin and soft tissue infections. Again, this is in the same poster session, and the conclusions of this particular poster, number B044, is the fact that polymorphisms in the genes for specific toll receptors actually increase your susceptibility for risk for complicated skin and soft tissue infection due to impaired toll receptor and pathogen recognition. Starting on tomorrow at 8:30, there is a new style of symposium in this category on malnutrition, the microbiome, and mucosal immunity. As we begin to look at the various factors that contribute to why certain hosts are more susceptible, we begin to look at the complexity of studying the environment, various host factors like nutrition, and the microbiome or the microbiology in certain compartments in the host. This is going to be an exciting symposium, I believe, and a very important talk is the characterization of the immune system of children in the developing world and how changes in the microbiome and their risk for malnutrition contribute to their susceptibility to significant problems. On Tuesday at 10 o'clock, there's a symposium, and there is a talk within this session that's entitled New Concepts in Tuberculosis, Pathogenesis, Diagnostics, Treatment, and Prevention. One of those talks is going to look at a recent contribution to the literature on B cells and the role of humoral immunity in tuberculosis. And in our past thinking about how the immune system deals with tuberculosis is that it's mainly the acquired immune response through T cells that is really the controlling factor. And here we're looking at the role of the humoral immune system in the control of tuberculosis. Moving on to a slide session at 10:30 on Tuesday, we're going to be looking at how certain treatments are being augmented by the use of new antibodies to particular proteins or molecules on a particular bacteria. In this case, B1735 on Tuesday at 10:30, we'll look at the efficiency of human immune globulin for the treatment of lethal necrotizing pneumonia caused by hypervirulent community-associated methicillin-resistant Staph aureus. As you know, treatment of MRSA is often quite difficult, and the use of novel antibody therapies for difficult infections is becoming a key hotspot. And in this one poster, B1735, they look at how IVIG is actually superior in their study to vancomycin for the treatment of necrotizing pneumonia using a rabbit model, and may lay the groundwork for future studies in humans.
I think that's a winner of one of the Program Committee Awards.
And it is. It's also one of the winners. B1736 looks at anti-alpha toxin monoclonal antibody reducing community-acquired MRSA disease severity, again in an animal model, but these are very important studies in laying the groundwork for future human studies. Again, this is Tuesday at 11:45 in a slide session in the Esplanade Ballroom, number 310. And this study actually, along with another, looks at how antibodies to alpha toxin, which is an important cytolytic toxin of Staph aureus, can improve and alleviate disease by MRSA. And the hypothesis here is that this is actually happening not by direct action of the antibody preparation, but by immunomodulation of the inflammatory process, resulting in accelerated bacterial clearance and skin healing. There's another to follow in this slide session, number 1737, looking at novel antibody therapy, again, of difficult infections, looking at the combination of anti-alpha toxin monoclonal antibody therapy with vancomycin in a mouse pneumonia model. And here again, they start to show that treatment with the combination is more effective than monotherapy, either with the anti-alpha toxin itself or with vancomycin. And therefore, again, we're starting to see signs that combining biologics with antibiotics may be the way of the future for treatment of certain types of infections. I want to move on now to the topic of pediatrics and vaccines. There is a very important slide session on Monday at 2 o'clock looking at vaccines, innovations, impact, and safety. One of the titles there, G1049, focuses on nasopharyngeal carriage in healthy children before and after receiving pneumococcal conjugate vaccine. We are trying to learn a lot more about what happens when you give vaccines that have multiple targets and its effect on certain disease pathways. And in this case, there are three abstracts within that particular slide session that are very important in evaluating vaccine impact on respiratory infections, specifically related to the use of pneumococcal conjugate vaccine. In all of these, they begin to explore how the vaccine actually impacts the different strains of pneumococci that colonize, the impact on acute otitis media, and the reduction of respiratory disease in those children. So I encourage you to go there because we are learning what the impact is of using vaccines that have multiple targets within them. Next area that I want to focus on is global pediatrics. And again, on Tuesday at 12:15, there is an important study looking at the contribution of socioeconomic and racial disparities on risk of invasive Streptococcus pneumoniae infection among children, and this is in a cohort in Atlanta. Although we have identified racial disparities as being well described for invasive pneumococcal disease, little information has existed up till now on how race interacts with community socioeconomic factors. And this study actually takes a good look at that and begins to note that socioeconomic disparities were noted for invasive disease independent of racial differences, and that vaccination appeared to reduce socioeconomic disparities in the rates of invasive disease by pneumococci in the whole population. And that among black children, worsening socioeconomic conditions were actually associated with lower invasive pneumococcal disease rates, while the opposite was seen in the total population. So we're beginning to understand some of the complex interplay between socioeconomic factors, race, and impact of vaccination. There's some exciting new activity in vaccine innovation starting on Monday in room 305. And my next three recommendations focus on that. The first one is on number 077, presentation number 575, looking at the development of a novel multi-antigen vaccine to prevent Staph aureus infection and disease. And this one looks at various new vaccines against Staph aureus that are coming down the pipeline. The second is presentation number 576, a universal influenza vaccine. And there's been more recent activity in the scientific community to try and develop a broad cross-strain vaccine against influenza. And I think that this particular session will actually target what's happening there. And lastly, a vaccine for HIV-1, something that has plagued our ability to develop over several years now. And this particular investigator will be discussing what's come down the road, what some of the challenges are, and also the new information on the isolation of broadly neutralizing antibodies, and how these are going to be used to design immunogens to induce similar cross-reactive antibodies in the form of a vaccine. Thank you.
Great. Thanks very much, Craig. Laurent?
So I will start with HIV. HIV, you will get the opportunity to assist to 3 symposia, 043, 152, 201, that will cover different fields of HIV. First, new antiretroviral therapy, new drugs, and new development in the field, but also what is most important in the field at this time for patients that are treated is the management of side effects, bone, metabolism, and also renal side effects. So we'll get a lot of issues here that will be discussed during this ICAAC concerning the management of these patients. During the slide session, there is also one late breaker that might be of interest. It's 556B, which describes the result of a phase 3 studies using a new integrase inhibitors that can be given once a day. This drug is called dolutegravir. There is also a paradox that is always there. If you go to session 126, it will be this week, I don't remember when. The session will discuss opportunistic infections in HIV, which are not the prime time here in the US or in Europe, but are still a very important issue in Africa or in Asia. And you will get discussion on TB, crypto, and other opportunistic infection. Let's move now to hepatitis. This morning you had already an overview and flavors on the development and the recent progress on hepatitis C. But you also add the comments that all patients could not benefit of the drug depending on the genotype, or also for many of them, given the side effects of the drug, many physicians decide to not to treat patients that don't need absolutely the treatments, although theoretically you can eliminate the virus. This issue will be discussed in session 034 and 039, and also in the field of transplantations, whether these new drugs could be used in the field of transplantation or not. Among emerging challenges in the field of hepatitis is also hepatitis E. Hepatitis E has been recognized as a cause of acute hepatitis, but also in immunocompromised patients as a cause of chronic infection. This challenging issue will be discussed Monday during Symposium 080 at 11:15. What about new antiviral therapy not related to HIV or hepatitis? During Tuesday for the Cubist Award lecture, you will get an overview from Larry Corey that describes the progress that has been done in the field and the progress that are ongoing and the future prospect in this field. It will be Tuesday at 4:30. Concerning new anti-herpes therapy, there is two communications that probably are of interest here. It's T356 and T362. These two abstracts during the poster session or the oral session will describe first letermovir. Letermovir is a drug by a small German company that has developed a drug that seems quite effective against CMV, particularly in transplant recipients. Another drug of interest that we already discussed last year and is still in development with new clinical data is CMX-001, which is an oral drug, which is also a lipid derivative of cidofovir, and you will get new data concerning the development of this drug. And then for those that are interested in resistance, since as soon as you have antiviral drugs, you have also resistance, there will be a symposia that will cover all the field from HIV to influenza, including hepatitis, on management of resistance in patients that receive antiviral drugs. This will be Wednesday, Symposia 235. What about central nervous system infections? You know that we are using more and more for multiple type of disease biologic agents. And these biologic agents have downsides, and one of the downsides is to reactivate or to promote central nervous system infection, GC, for example, and other viral infection. This issue will be discussed not only during a symposium, which is number 122, Monday at 2 p.m., but also during other different posters or oral sessions. I can mention, for example, for those that are interested, the poster T342, that described the reactivation of our disease in patients with multiple sclerosis treated with these biological agents. And finally, a symposium that I think is very interesting, not only because it discussed important issue in terms of central nervous system infection, viral central nervous system infection, but will describe also from an Asian speaker the recent very, very large outbreak of enterovirus 17.1 in Asia that many of us does not know, but that has caused really thousands of kids and young babies that have been admitted in intensive care units across Vietnam, Cambodia, and other countries for brainstem encephalitis following enterovirus 71 infection. 2 words concerning viral infection and transplantations. There will be several sessions discussing CMV control in this patient, which remains important issues. This will be discussed during a slide session 116, that will be Monday afternoon. And you will get also, for those that are interested in transplantations and clinical aspect of transplantations, a very interesting interactive session Monday afternoon from 4 to 6. And let's finish with flu. There is one session on flu that will be 195, Tuesday at 11:15. That will raise several questions. First, do we need to care, still we need to care about H5N1? Is there still an issue? And another point, we will get a representative from the CDC that will discuss the recent H3N2 swine influenza virus outbreak here in the US with more than 250 cases, and discuss whether this needs to be considered as a serious issue and whether there is already some human-to-human transmission. And finally, a quite provocative late-breaker abstract on flu, which is V400, that describes that in animal models, in this case ferrets, when ferrets are vaccinated with an inactivated trivalent vaccine and then challenged with H1N1, the severity of disease is increased. This just tells you that vaccinations can change the face of disease, can prevent disease, which is the first goal of vaccination, but also we have to learn when it changes disease in a way that we do not expect. So it will be late breaker abstract V400. Thank you.
Thanks very much, Laurent. So it's now my task to cover clinical issues at ICAAC, and I thought I'd just go through day by day. So there are actually two posters that are on currently, which I think are quite notable, and they're on from 11:30 through to 1:30. The first of these is L2-301, and this is the winner of one of the Program Committee Awards. It's entitled Prevalence of Vaccine Preventable HPV Infection amongst a cohort of men who were being followed for anal carcinoma, and essentially shows that about 50% of the anal carcinomas found in this large Sydney cohort actually had HPV or papillomavirus strains that could have been prevented if they had been vaccinated with the HPV vaccine. So at a time when a lot of countries are looking at issuing HPV vaccine for both boys and girls, so boys in addition to girls, this is an important study and is a winner of one of the awards. That's 301. And also at the same poster session, L2-308 is genetics and molecular epidemiology of cephalosporin resistant Neisseria gonorrhoeae. So many of you may have noted that the MMWR recently reported that cefixime was no longer active against gonorrhoea, and there have now been a number of reports of ceftriaxone resistant strains, and this is an interesting poster looking at the epidemiology of that here in California. For this afternoon, from 4 till 6, there are two symposia you might want to pay attention to. One is Symposia 037. It's looking at multi-drug resistance in emerging countries, particularly India, Brazil, South Africa and China. And of course, with the emergence of NDM, this is particularly notable and features quite prominently in that session. So these are critical areas, and of course, some of you be aware that there's recently been NDM reported in Africa, in Cameroon and other places without transport links, if you like, to India and the Indian subcontinent. So this will be an important session. And also Symposia 044, a change of topic, but an update on novel and important tick-borne diseases, not only covering Ehrlichia, but also a very interesting session on the deer tick virus that is becoming a problem in parts of Connecticut. For Monday, there are a number of sessions. Firstly, Symposium 061 at 8:30 to 10:30 is a session looking at polymyxins and their activity against gram-negatives. Clearly a huge issue, problems with nephrotoxicity, dosing, but also in this session you'll see looking at what to do in patients of different body weight and so forth. So at a time when many parts of Southeast Asia, for instance, are now using polymyxin as part of their empiric drug therapy when patients are first admitted to ICU, many of the issues that are being discussed in this session are very, very relevant. At the same time, this is Monday 8:30 to 10:30, Symposium 069 covers new issues in the management of MDR and XDR tuberculosis. And you'll see the speakers in that session really are the cream of the crop in terms of their expertise in XDR-TB. We have representatives from WHO, but also Bill Bashai, who has a large study going on in South Africa, an incredibly important symposium. At lunchtime tomorrow, or not quite lunchtime, but 11:15 to 12:45, Symposium 078 is a review of 50 years of fluoroquinolones. So this year is the 50th anniversary of the fluoroquinolones, and there are 3 speakers looking at the emergence of resistance to fluoroquinolones, the new use of fluoroquinolones outside the field of gram-negatives, and then David Hooper, who's really one of the world's experts on this area, looking at the new fluoroquinolones that are coming down the pipeline. In the afternoon, there is an important poster session. And for those of you who missed this morning's talk about MALDI-TOF and new technologies in the laboratory, there's an important poster session entirely on MALDI-TOF technology, session 090 from 11:15 to 1:15. And that's an important one to get to. There are too many abstracts to talk about individually now, but to give a good idea of how this new technology is changing clinical infectious disease, it's a good session to go to. Of course, we can't leave Monday without mentioning ICAAC Live. Vincent Racaniello and Mike Schmidt will be chairing this session, and they'll be interviewing live John Brownstein, Bill Bashai, who I just mentioned is an expert in TB, and Victor Nozay. That's from 2 o'clock till 4 o'clock in Esplanade Ballroom 203. From 2 till 4, competing with this, Symposium 127, which looks at new issues in the management of XDR or MDR endocarditis. Now, this is not a topic we would have ever bothered to discuss up until the last 3 years or so, and increasingly we're seeing problems now with multi-drug resistant pathogens causing endocarditis. So to cover the various areas, staphylococcal disease, Chip Chambers, enterococci, Bob Mollering, gram-negatives, David Patterson, and then some of you may be aware that in parts of Europe, particularly the Netherlands, they've had major problems with Q fever endocarditis, and so there's a talk from one of the experts from the Netherlands on Q fever, Corrine Delsing. And then on Monday evening, 4:30 to 5:30, of course, is the ICAAC lecture. Arturo Casadevall, a true giant in the field of fungal therapy, particularly Cryptococcus neoformans, will be talking about fungal disease in the 21st century. For those of you who speak Chinese, we actually have a new session this year after the ICAAC lecture, purely for Chinese delegates, where there'll be 4 talks that have already been covered in ICAAC, but where Chinese delegates can come and ask questions in Chinese, and there'll be translation in Chinese. My Mandarin's not quite good enough, but we will have people who have better Mandarin than mine to cover that. For Tuesday, the 11th, so in the morning, 8:30 to 10:30, there are a number of sessions. Symposium 147, vancomycin, what do we do about PKPD? There's always a session about vancomycin, but I think this session is particularly notable because although we're always looking for new drugs, of course, the other alternative is to the dosing issues with older drugs such as vancomycin, and a number of the speakers there particularly looking at nephrotoxicity, but also the impact of MIC shifts to vancomycin, particularly among staphylococci, are 2 interesting talks in that symposium. At the same time as Symposium 153, that's on at 8:30 to 10:30 on Tuesday. For shift in gears here, if you like, dangerous pets and other animals. And many of you may not be aware, but it's now emerging, in fact, that many of the zoonotic diseases that are a real problem are actually spread by bats. Bats are increasing their role in the ecosystem, incredibly important in terms of fertilizing flowers and trees and so forth, but also potentially in spreading viral diseases that they themselves are not injured by, but which can then enter other animals. And the first speaker in this session, Dangerous Pets and Other Animals, Dr. Linfar Wang from CSIRO in Melbourne. He has been the key behind investigating the Hendra virus outbreak in Australia, but also the Nipah virus outbreak in Malaysia, along with Malaysian colleagues, of course, and is an expert, as he calls it, in his bat pack, instead of the rat pack, but it gives a very interesting talk about the role of bats in the dissemination of zoonotic disease. Also in that session is Ian Lipkin from the Green Lab in New York, a real leader in terms of deep sequencing and identification of new viral pathogens. A talk on MRSA and animals, and particularly pets. And then finally a talk about animal bites and the problems with that. Competing with that session is Symposium 154. This is Tuesday again at 8:30 to 10:30, what every clinician should know about emerging new antimicrobials. And the purpose of this session is 5 short, sharp talks about new antimicrobials that have come on the scene in the last year or two, or reviewing updates such as for drugs such as ceftaroline, which have been increasingly in use over the last couple of years. So that's an important session to attend. I think the other notable post or talk you might wish to attend is C2-1219. This is on at 10:45 on Tuesday morning. And it's the first report of NDM E. coli from companion animals in the US. So, of course, we've seen problems with MRSA with companion animals, but this is the first report of NDM E. coli, which is obviously of major concern. So that's an important session. And at the same time, there's a separate individual slide presentation in between 8:30 and 11, L1-1221. Is CSF lactate the best parameter for discriminating between bacterial and viral meningitis? And this was one of the winners of the Program Committee Awards this year in the clinical field. And there's a really notable study from Lausanne, Switzerland, suggesting that CSF lactate, which is not something we usually measure, is a very important predictor of whether or not you have viral meningitis or bacterial meningitis, and far more useful than measuring white cell count, glucose, or protein, which is all the traditional things. So this study is a potential game changer clinically in terms of community-acquired meningitis. In the afternoon, slide session 201 from 1:30 to 4:30, there are 2 state-of-the-art mini lectures that you might wish to attend, both on new technologies in the laboratory about MALDI-TOF and PCR and ESI mass spectrometry. So if you're wanting to just go to 2 sessions about how to update your knowledge about new technologies, these would be the 2 to go to, the one I mentioned earlier. And then this. And then at 2 o'clock to 4, the ID quiz. I've seen the slides this year, and some of the questions are, I think, impossible to answer, but I'm sure someone will be able to answer them other than me. But it's always a bit of fun and a good way to test your knowledge. Followed, of course, by the Cubist lecture, which is Dr. Laurent Corey, who's a real giant in terms of antivirals, particularly anti-herpes therapies, and also in terms of setting up many of the ACTG networks that are currently underway. Craig will be introducing Dr. Corey. So that's Tuesday, and then finally on Wednesday, 3 symposia you may wish to note. Symposia 225, 8 o'clock to 9 o'clock. This is a plenary session, in fact. It's the first time we've had a plenary session on the Wednesday. And this is very notable because the topic is on the use of social media and new technologies in healthcare. And this is a really rapidly emerging field. I had dinner with Neil Ferguson, one of the speakers last night, and he was informing me that they now have, I think, $28 million of funding purely on this topic from the Gates Foundation. So Neil Ferguson from Imperial College London and John Brownstein from the Children's Hospital in Boston and Harvard Medical School, both of whom have done some of the cutting-edge research using mobile phones to track spread of interactions between people and then spread of disease and so forth. A very important plenary session, 8 a.m. to 9 on Wednesday morning. Then 2 final symposia, Symposia 232, What's New with the Enterococcus, important update on an important organism, and Symposia 234, Best Practices for Serological Testing in Selected Parasitic Diseases. Not to forget all those important parasitic diseases, but there have been some very major advances in that field, and there's a symposium specifically on that. So at that point, Jim, I might stop and throw it open to you for questions, to anyone on the panel.
Yeah, just a reminder for those of us who are watching over the internet, If you have a question, please submit it via Twitter using the hashtag #ICAAC or directly at ASM Newsroom. Otherwise, if anyone in the room has any questions, please raise your hand and I will bring you the microphone.
We've answered all their questions, Jim. There's none
Apparently
left.
so. Apparently so. Otherwise, I want to thank the members of the program committee for coming today to give us an overview of the program. This concludes the overview briefing. Our next briefing will take place at 1 o'clock Pacific time on measles.
Explore the fundamental role of microbes in the natural history of our planet with Microbes and Evolution, the world that Darwin never saw. Available at estore.asm.org.